my dear viewers, I welcome you all to my class. I am Ekta Singhvi, your biology teacher. Currently, we are discussing classification of living world. In this context, I have already made one video in which we have discussed the overall scenario of the classification, and in this video we shall dive into the groups, the different groups that are in the classification. We currently follow 5 kingdom classification given by Robert H. Whittaker, and according to this 5 kingdom classification, the first group belongs to Monera. Monera or the prokaryotes. After this we have Protista, then fungi, then plantae, then animalia, that we shall be doing subsequently, but in this video we shall be chiefly discussing about Monera. So let us see what monerans are. First of all, I would request you to please go through this slide, this board work, and if you can, please note it down in your copy, because these are the most important points so far as monerans are concerned. So, what are monerans? Monerans are prokaryotes, whom we know commonly as bacteria. These are the most primitive cells of the living world. When the world was being formed and when first life came up, around 3,500 million years ago, that was Monera. Monerans are the ones which can survive in very difficult situations like oxygenless, or where it is very saline, or where temperature is very high, say 100 degrees centigrade. In all those situations, monerans can survive. These are living beings, these are the pioneers of life. They are omnipresent, and monerans, they are so delicate that they cannot be classified on morphological basis or their structure. They are classified based on their biochemical, physiological or ecological entity characters. So let us see what the classification is. So they are divided into 2 groups. First is Archaebacteria, which is more primitive, and they are obligate anaerobe. That means they cannot survive in oxygen. They lack peptidoglycan, which is the basic building block of bacterial cell wall. These bacteria, Archaebacteria, do not contain peptidoglycan in their cell wall. Rather, they contain pseudomurine. So this is a very important distinguishing factor of Archaebacteria and eubacteria. Archaebacteria are more primitive than eubacteria, and these are the primitive qualities. Archaebacteria is again divided into 3 divisions. They are methanogens, halophile and thermacidophile, depending on their qualities. Like methanogen, they are able to produce methane. Halophile, they are able to sustain very brine environment, very saline environment. And thermacidophile, they can live in even 100 degrees centigrade, like hot spring and all. Thermacidophile can sustain. So this is what Archaebacteria is. Now let's come to eubacteria. Eubacteria are the true bacteria. True bacteria in the sense that the cell wall contains peptidoglycan. Peptidoglycan is a chemical which is found only in bacterial cell wall, and that is what true bacteria has. They are facilitate anaerobes. That means they can survive both in oxygen or without oxygen. With oxygen and without oxygen, they can survive in both conditions. And eubacteria is again divided into bacteria and cyanobacteria. Bacteria and cyanobacteria. So basically in bacteria we have got these 5 classifications, and this is how they have come up in the world. That means Archaebacteria are the oldest one, after that came bacteria, and after that came cyanobacteria. That means cyanobacteria are the most recent type of bacteria, and they are rather more close to algae than to bacteria. But because their cell wall had peptidoglycan, they were brought under bacteria. Earlier, cyanobacteria was classified with algae because they had lot of qualities which resemble algae. First of all, they have chlorophyll, or any of these groups, they do not have chlorophyll. Bacteria have bacteriochlorophyll. They cannot release oxygen. But cyanobacteria are the one which can release oxygen, and thus they became the first harbinger of life all the different varieties of life that came after cyanobacteria, because cyanobacteria filled the world with oxygen. Earlier than cyanobacteria, the atmosphere of Earth was anoxic. That means there was no oxygen in the atmosphere. It is because of cyanobacteria that oxygen has come, and so we all should be thankful to cyanobacteria. Now let us see the characteristics of all these different varieties of bacteria. Methanogens are a type of Archaebacteria, and they can produce methane from the organic compounds. They are found in the stomach, in the rumen of ruminants like cow, buffalo, goat, etc. These animals, they have 4 compartments in their stomach. The herbivores which live on the plants, they all have 4 compartments in their stomach, and the first one is known as rumen, in which cellulose, when it goes, there are methanogens present there, and they digest the cellulose, and as in doing that they produce methane gas. So they are found in stomach of ruminant, and they can produce methane, they can digest cellulose. Since they can produce methane, they are used in production of biogas. They are used in sewage treatment, from where methane is produced, and methane is used to produce biogas. Example of methanogen is methanococcus. Then we have thermacidophile, which is yet another type of Archaebacteria. Thermacidophile, they are found in hot spring, and they are also highly useful in biotechnology, because the enzyme that they contain, that they do not get destroyed in high temperature, and as such they have got lot of use in laboratories. For one, they can be used in PCR, polymerase chain reaction, in which from one single strand of DNA we can produce million copies of DNA, and that is done with the help of Taq polymerase, which is an enzyme which is found in Thermus aquaticus, which is a type of thermacidophile. Besides this, they have got many other enzymes. One example would be restriction enzyme. These are used as molecular scissor. They can be used to cut DNA in some particular places, and they can be used for further experiments. So Archaebacteria, they are highly useful in biotechnology. Our next group is eubacteria. Eubacteria means true bacteria, which has cell wall made up of peptidoglycan. Now, if you are asked difference between eubacteria and Archaebacteria, you can mention 2 points. First, eubacteria can survive in oxygen-rich atmosphere, and secondly, their cell wall has got peptidoglycan. Eubacteria is again divided into 2 branches. One is bacteria and the other is cyanobacteria. Cyanobacteria are bigger than the normal bacteria, and they can do photosynthesis. Yes, cyanobacteria are the first organism which could do photosynthesis. Before this, all the organisms that were present, bacteria and Archaebacteria, they did chemosynthesis. They couldn't release oxygen into the atmosphere, but cyanobacteria could release oxygen into the atmosphere, and they made the presence of oxygen in the atmosphere, by the help of which all the organisms entered. So cyanobacteria are responsible for so much of extension of life, so much of proliferation of life. We should be thankful to them. So they contain chlorophyll A, which is present in plants and cyanobacteria. They also have phycobilin, because of which their color is typical blue-green, and so they were included in blue-green algae. But since it was discovered that their cell wall has peptidoglycan, they were shifted to the bacteria group. They can do nitrogen fixation. Sexual reproduction is altogether absent in this bacteria, and they are the important source of single cell protein, which is so much useful in present day. It is used for fodder, it is also used for human consumption, single cell protein, and it is derived from spirulina. Spirulina is a type of cyanobacteria. Other examples are Anapina, Geocaspa, Nostoc, and so on. All these varieties, they can do nitrogen fixation, and so they are quite helpful in the environment. Even now, maximum photosynthesis is being carried out by cyanobacteria. So this is what their cell has important structures like heterocyst, which are resting spores. They have echinate, Echinates are the resting spore and heterocysts are for asexual reproduction. So these are characteristic feature of cyanobacteria. They are also found in algae. So a lot of features of cyanobacteria resemble that of bacteria. A lot of features resemble algae also. So they are supposed to be a connecting link between bacteria and algae. So that's it. That is how evolution happens. So with this we come to an end of this video, and after this we shall be discussing bacteria. Please keep following the video and please keep noting down the points, because these points are very important for you all. Thank you so much.